Okay, I think we're ready to get started now. It's a great pleasure to welcome you here today, and grande piacere di averti qui. And I normally broadcast these from my library at home, which is where we are here, but I wanted to make you feel very comfortable, and so I'm going to Zoom to a different library now.
The fresco room.
Do you see that where we are?
I recognize that's my room.
It's your room, I know it is. I put this up here so that I could show the contrast between between your library and the library that we have in our small little wooden building here on the coast at the Oregon Institute of Marine Biology. For those of you who don't recognize it, this is the fresco room at the Stazione Zoologica di Napoli in Naples, Italy, where Roberto is the director and the president of that organization. And these frescoes on the wall that surround one of the rooms, one of the many rooms of the library, were done in the 1880s, I think, by a famous German artist, and they depict some of the early days of fishing and so on in Naples. It's a spectacular place where Roberto works. And I'll just put up a different picture of the library as well as a background. This is in the same room to give you an idea of what the place is like. So let me introduce Roberto Danovaro. Roberto is one of the very prominent marine biologists, or biologists in general, in Italy and in Europe. He began his work at the University of Genoa, where I believe he did his master's degree, and then moved to the University of Pisa, one of the very old universities in the world, really, and got his PhD at the University of Pisa. He's been a professor for many years at the University of Marche and Polytechnic University of Marche, and he's worked his way up through the ranks there, through the professorial ranks. I think he's been the dean and the rector, all sorts of things, very high level in the university, and was chosen a number of years ago to be the director of the Stazione Zoologica. This is a place that's really important in the history of marine biology for you students who don't know this. This was really one of the very first marine labs in the world, and I think it's the oldest one that's still functioning. And it was founded in 1872 by a German, because Germans don't have much of a coastline, and they used to come to Italy, and especially in the wintertime, to work in various places. And Anton Dohrn, a wealthy man, supporter of Charles Darwin, founded this place, and it quickly became a place of international importance that welcomed people from all over the world to come there and work at the benches, the national benches that they opened up. So Roberto is the president of that place. It is still an extremely important place for research. And what else can I say? Roberto has been very productive as a scientist. He works in a lot of different areas. He's perhaps best known for his work on conservation biology. He's worked in conservation biology in both shallow water and the deep sea. As you heard a few minutes ago, he has a very broad view of conservation for the world, for the planet. He also is an expert on viruses, and he's an expert on invertebrates for that matter, and has worked in everything from aquaculture to deep sea biology, written 3 books. I'm very familiar with one of his books on how to handle deep sea sediments and how to interpret deep sea sediment biology. Worked in lots of places, from the Antarctic, various oceans, lots of oceanographic cruises. One of the things I really like about Roberto's record is that he has a number of important awards and honors that have been given to him. One of his papers was chosen by Biomedical Group up in London as the best biology paper in the world back in 2010. which I suppose by inference means that Roberto is the best biologist in the world, right? At least he was in 2010. Okay, he's denying that point. I was also impressed that he got the French Oceanographic Prize a few years ago and was presented that prize in Paris by the Prince of Monaco. So there's some nice pictures of that event, I'm sure. Well, I think we've done enough to your ego now. So he's published about 300 papers, about 3 books, I think, and very, very productive. Lots and lots of citations. Has a citation record that would make most of us very jealous. So with that, Roberto, I will turn the time to you, and thank you for being here. We'll hear from you now about whatever you want to say.
Too kind of you. Thank you, Craig. Thank you for your presentation. You said far too much because you know that the long list of things you mentioned just indicated there's a long time and I work in this sector because I started very early. I got my degrees when I was 22 and I finished my PhD at 26 and I started the career at the University at 27 and that was ended at 33. So the point is that it's a long time. It's 20 years that I completed my career at the university. So you need to expand, you know, your fieldworks. And I suggest for the students, and I'm going to share in a second the first slides, is that I never thought I could become an expert in viruses. I was, you know, just approaching the marine life because my father was just fishing and I was, you know, going to sea and I was loving the nature and I love, you know, crabs and you know, the things. And so I approach it in this way, you know, like everyone, you know, that loves dolphins, loves big things. And then my professor said, well, look, there is you know, an invisible majority that can be of interest too. And I discovered that indeed, you know, the microbial world was, you know, almost unknown at that time, at least when I started my PhD in 1990 or so. And the nice thing was that when I started my PhD, I was rejected because they say, well, are you sure microbes are present in the marine environment? Not sure about that. In any case, you can try, but you repeat another year. So I had to propose again my PhD saying, look, I'm convinced that there are a lot of microbes there. Let me demonstrate it. So that was, you know, it looks like you know, in the Pleistocene. In reality, it was just some 30 years ago. So a lot of things happened since then. And thanks to the invitation by Craig, I'm going to make just an overview of some of the recent advancements on the microbial components with special emphasis on the invisible majority represented not just by bacteria or archaea, but also by viruses. Is a very timely topic considering that the COVID pandemic is related to a virus. So tiny particles that are affecting human life so strongly. So the outline of the talk is looking at the importance of the different life forms, especially in the dark, because this is an important ecosystem, as I will show you later on, you know already. So the role of viruses in deep ecosystems, in the function of the global biogeochemical cycles, the role of viruses in creating a sort of soup of life all over in the blue planet, especially in the deep sea. A glimpse into the present knowledge of viral diversity and the potential interactions of this diversity with the functioning and productivity of different other components, and then also a look at the effects of global climate change of viruses and implications for the functioning of the global biosphere. So we all know that we are looking to the largest biome of the biosphere, the oceans, but particularly the habitats beneath 2,000 meter depth are technically defined deep, truly deep. And these represent likely more than 95% of the biosphere. So we need to consider the 3 dimension of the oceans and look at the water column down to the sediments. We all know that still we know too little about the deep ocean in particular. And this is due to the technical constraint, the need of advanced technologies we know maybe we map at 5% of the oceans. Looking at the tiny, well-known sea like the Mediterranean Sea, we still have no complete bathymetric map of this small ocean that represent just 0.8% of the ocean surface. And this is the same everywhere. I was, just crossing the Pacific from Chile to the Easter Island, and we discovered just a couple of seamounts and whatever. So most of the habitats are unknown, so not mapped. Just the yellow part is mapped, the rest is not mapped. And the component we know, we recently also estimated in a paper published with Paul Snellgrove and Paul Tyler on transit ecology evolution, is as much as 0.001% in terms of amount of sediment surface that's been explored in terms of biological components. We know that life is present all over the 3 dimensions, and of course is present across the entire water column, across a depth of on average almost 4 kilometers. It looks like the average depth is 3,750 meters actually. But then when we touch the bottom, when we touch the sediment surface, we still have to dig into the sediment, and we can find life, large forms of life in the top, maybe 1 meter of the sediment, but then we can go for maybe 1,000 meters, finding other life forms such as bacteria, archaea, and viruses too. Another issue that has been always overlooked is the fact that we don't know when we talk about the ocean seafloor, that the ocean seafloor is not just the orthogonal projection of the surface of the oceans. It's much more than that, because there are slopes, there are domes, there are seamounts, there are ridges and so forth that increase the overall surface of the sediments. That is actually all over the world, the sediment represent an hotspot of life and biodiversity. To be honest, it's an hotspot because we investigated better, far better, the sediments that the water column. Possibly the very last frontier is not just the deep sea, but is the deep water column of the oceans, even more than the sediments. But despite all these, consider that the actual surface of the ocean is about 374 million square kilometers. The actually bioavailable benthic area is as much as 434 million kilometers. So it's about at least 20% more than we estimated so far in terms of surface. And this is important because when we make projections about the global number of life forms there, the global biomass, or the biogeochemical cycles or whatever, we always consider just the orthogonal projection and not the actual surface on which biological processes occur, on which life is present and colonizes. So we started a few years ago looking at the viral components. Of course, this was almost impossible till the 90s, where there were not available the techniques related to the, for instance, epifluorescence microscopy, the ability to stain using fluorochromes, either the DNA or the RNA of prokaryotes or viral particles. So there was a lot of uncertainty about that. So the first step was in the 90s to set, solid, robust methodological approach to quantify these components and to confirm that also from the point of view of the most, minuscule forms of life, like bacteria, archaea, and viruses, these are present all over the world. And this was somehow expected because the azoic theory, of forms was, already demonstrated to be a fake since many, many decades. But the assumption was also that every form of life, even though present till the deepest depths, was decreasing exponentially with increasing water depth. if you look at these graphs and you look at the viral abundance, for instance, you might appreciate to have that at a depth of more than 5,000 meters, the viral abundance can be as much as in most, coastal or relatively shallow systems. So there is no clear identifiable pattern for viral abundance, and little evidence also of any pattern for viral production. This means that these components are not just surviving by a rain of particles settling down through the water column and reaching the seafloor and then maybe degrading or disappearing, but these components, since they produce all over the sediment surface at any depth, they are active. So they are doing their own work. They are infecting components. And when we look at the hosts of these viruses, it is evident that it's not just the microbial world. Of course, the number of microbes is huge, so most of the viruses infect bacteria and archaea. But we know very well that all of these viruses infect any life form, any invertebrate or vertebrate at any depth. So these are very active components. Viruses can reproduce. They can have a viral turnover on the range of even 30 hours or so, even at abyssal depths. So we tried to recalculate a little bit the numbers and understand what is going on for the different components. And looking also to the patterns of abundance in deep sea sediments. There are, keystone papers published by Rex for many years demonstrating these patterns. However, the point is another one. When you look at the abundance per square meter, you look at an exponential pattern. But when you look in this case at the overall abundance for any bathymetric depth, so counting all, for instance, meiofaunal organisms present in the bathymetric, 1,000 to 1,200 meters, plus those present in all sediments in the bathymetric 1,200 to 1,400 meters, and so forth. You understand that it is true they decline per unit of square meter, but since increases the surface area of deep sea sediments, and the depths are at 4,000 meters are far, wider than those at 200 meters, you discover that the overall abundance in these deep sea sediments is higher than at shallow depths. So it indicates clearly that maybe more than 95% also of the life, even the microbial forms of life, are in the deep sea and not just in the shallow. Of course, the trend is less evident for large components. If it increases for the microbial components, for instance, for bacteria and for archaea, it is almost constant for every 200 meter of bathymetry. For meiofauna, slightly decreasing for macrofauna, decreasing more substantially for megafauna. But still, if you compare the abundance estimated for the top 200 meters or so, and you compare with the overall abundance in the rest of the sediments, you get, in any case, the same figure. So most of the life, by far, is in the deep sea and not in the shallow. So, given that premise, why it is important to include viruses in our studies and our modeling of our cell processes? Given the fact that we have overall more than 10 to 30 prokaryotes, so that means bacteria and archaea that should be considered separately according to recent approaches, and probably more than 10 to 31 viruses, we have to keep in mind that they are playing a huge work in there. 1 kilogram of deep sea sediments can contain 10 to 12 viruses and 10 to 11 prokaryotes. So this is a potentially inexhaustible source of new genes when you just do, and it's evident in bioprospecting studies, you get just a bit of sediments everywhere and you do a metagenomic and a metatranscriptomic approach. you do a fantastic job in terms of gene mining and exploitation of potential functions, including new functions. But what is more interesting from my point of view is the ecological role. I'm not a microbiologist, I'm an ecologist, and I wanted just to explore the components that were, at least investigating in terms of ecological functions. So we culturally try to separate the microbiological approach to the ecological approach, like if ecologists have to work only in large components, see either animals, habitats, whatever, and microbiologists just on microbes, like the two components were independent. I'm convinced that even more in the future, we will understand the interactions between the two, like for humans, the importance of pathogens with respect to the human life and well-being. So viruses must be clearly integrated into global biogeochemical cycles for two main reasons. One is they exert a strong control on prokaryotes, which in turn exert overarching control on all biogeochemical processes. And second, because they also play a role in biogeochemical cycles, as I'll explain, and because they have been never taken into account in any ecological model. There is also an important issue in terms of evolution, because the viruses are at the end are some minuscule predators, and they exert a sort of predator-prey coevolution mechanism. So looking at the picture, at the global picture of carbon biomass and production, we can easily see that if you look at the first, 1,000 meters of depth, prokaryotes are there, but the most important component in terms of biomass is related to macrofauna, of course some megafauna, a bit of meiofauna, and some prokaryotes, but macrofauna is likely the most important components. But if you go from 1,000 to 4,000 meters, or from 4,000 to 6,000 meters or more, you see that it's progressively increasing the relevance of the prokaryotes, decreasing the importance, the quantitative importance in terms of percentage of the macrofauna, and of course the megafauna, and it becomes more important the component of the meiofauna. But viruses were not included in these global estimates. Look at the production. In terms of production, of course, the small things reproduce much faster. So we have that if we look at the global benthic carbon production, we made some estimates from our publications that the range in between 3.5 to 1 petagram of carbon per year globally, with important contribution of prokaryotic carbon production at all depths down to abyssal sediments and more. In addition, this is puzzling most of the deep sea scientists, because they say, well, how is it possible that just maybe 1% of the carbon production in the euphotic zone can sustain such an active benthic, community at all levels, including microbial components, that is working so fast, is reproducing whatever, without being food limited or being less food limited than expected. And here's the point of the importance of the so-called dark energy, the chemosynthetic production in the deep oceans, because there is an important component, not just limited to the archaeal component, but also including some bacterial components that carry on chemosynthetic production, thus increasing the carbon budget, the overall carbon budget, so providing more biomass to consume, to degrade, than that we expect by carbon export from the ocean surface. And it is difficult to make an estimate about that, but we know for sure that at least maybe for 20% of the carbon budget in the deep ocean, at least, is due to the contribution of this dark energy. And this is also confirmed by the fact that if we look at the relationship between heterotrophic carbon production and chemototrophic carbon production in the deep, we found a significant relationship between the two. That means that the two processes work together, and very likely heterotrophic carbon production can support chemototrophic production and vice versa. It is also evident that the viral lysis of some bacterial microbial components releases some components that are used by other microbial components, heterotrophic bacteria, that are not killed by viruses. So in other words, viruses play in the deep sea the same role of lions in the savannah, where they just kill components to make alive and productive and healthy the population of their prey. Some 10 years ago or more, we were also trying to understand better the impact of viruses in this world that was progressively more dominated by bacteria and by archaea than with respect of shallow sediments. So since the 90% of the global biomass in the deep ocean, in the deep sea sediments, is accounted by bacteria and archaea, or prokaryotes in general, the role of viruses should be prominent there, because they have, a special role in being almost the only components limiting the growth of the microbial components or the other microbial components. If you reduce the number and the biomass of the posi feeders and all other organisms that can consume, can uptake, can use also microbial biomass, other biological components such as viruses will play the preeminent role for controlling their growth. And this was clearly demonstrated by the study we conducted in Olosas that got also the cover of Nature, was accounted on top 20 paper in Nature that year. So it is demonstrated that viruses are virus-induced prokaryotic mortality, not just increases with water depth, and accounts for more than 80% of the shunt of the prokaryotic production in the deep sea. So it is kind of soup where microbes grow and viruses kill, and they circulate at much faster rates, all organic components in there. We also wanted to understand a little more of these interactions and distinguishing between the lytic viruses from lysogenic forms, so the viruses that were just killing, with respect to those that were just infecting without killing, maybe preventing also other infections, and so forth. And we demonstrated that there are either prokaryotic viruses and eukaryotic viruses. Of course, eukaryotic viruses represent an important component there, because it's evident that there are not just bacteria to infect. There are also protozoa and multicellular organisms. And we were even surprised by the relative importance of eukaryotic viruses that was much higher in proportion to the abundance of individuals. It means that, like, if larger organisms have a much broader range of viruses and broader range of potential disease to which they are susceptible. We also try to investigate a little bit more on the virus host range and look at the percentage of viral proteins per host kind in different environments. And we were also able to demonstrate that, of course, an important fraction of viruses has as a primary host bacteria, but an important fraction as also archaea or others, plants. Maybe they were just exported by microalgae and detritus falling down across, along the water column. And so for animals, very important fraction of animals infected, viruses infecting animals, and also anaceae components. After that work in nature, we wanted to understand a bit more and distinguish in between the role of viruses in infecting archaea and bacteria, because we we were just quantifying them through methodologies that since 2008 were unable to distinguish in between the two components. So we had to develop a new approach in there. And we found a new way to demonstrate the selective release, selective infection and burst of bacteria versus archaeal cell by looking at the genes that were released. So we were looking at the DNA or RNA that was released by infected cells, and using a quantitative real-time PCR, we were able to quantify selectively the number of archaea and the number of bacteria that were selectively killed. And it was very, very surprising that the impact on archaea was even stronger than the average impact on the world of prokaryotes, in particular than on bacteria. It's like if archaea, sorry, can you listen to me? I hope so. This is indicated that there are some problems of volume. In any case, I assume that it's fine. It was like if archaea were more evolved than, bacteria were more evolved than archaea and more able to contrast or to tolerate viral infections. In other words, archaea were killed at the rate, higher than bacteria. So viruses were preferentially killing archaea. This has implication, of course, also on the release of extracellular DNA. This was, fitting very well with a paper we published, Delano and myself, in 2005 on Science, where we demonstrated that more than 90% DNA present in the, there is not a problem with the, all right, should be okay. More than 90% of DNA present in the ocean was extracellular DNA, was not DNA within the living cells, it was DNA that was released in the environment and was remaining in the environment. So, the first conclusion is that viral shunt, viral infection, is a key source of labile organic detritus in the ecosystems. In turn, this contribution supports prokaryotic metabolism, stimulate prokaryotic growth, and also potentially contributes to the global models and cycles of carbon, nitrogen, and phosphorus cycling. And this was particularly evident in extreme ecosystems, as we publish also on ISME journal. Let me go a little bit deeper on this problem of extracellular DNA. I found very interesting this component because when I started my studies, I was looking at the biochemical components of organic matter in the oceans. And they are typically dominated by, so there is a bulk of carbon, largely refractory. Then there is a bioavailable components, lipids, proteins, carbohydrates, generally dominated either by carbohydrates or by proteins. And we were stopping there in the 80s and early 90s. And then we discovered that the fourth component in terms of abundance and concentration after proteins, carbohydrates, and lipids was where the nucleic acids was basically DNA. So DNA is also an important food source for most life forms, including large deposit-feeding organisms. So basically, the life of growth and infection and death was releasing, even by natural death of the organism, was releasing a lot of extracellular DNA, putting that in a sort of loop. Of course, viruses play a preeminent role in that release because they affect either bacteria and other organisms, and killing them, they release this DNA. And we wanted also to look at the sources of this extracellular DNA, understanding different sources, and of course, in the deep and everywhere in the world, the primary organic matter, the primary biomass, is a key component also of the flux of DNA into deep. Of course, there is also another component related to the biomass of organisms living into the sediments and contributing to the release of DNA in situ. But we were looking at this component, DNA in particular, because DNA is the molecule in absolute richest in terms of organic phosphorus. And phosphorus is a limiting component for all life forms, especially, but not only for the primary production. Looking at the role of extracellular DNA, but looking also at the abundance of viruses in the deep, we're guessing that, of course, we demonstrated on science that an important role in terms of phosphorus cycling at the global scale is played by the detritus, the extracellular DNA. A crucial role is played by viruses that they also die, and their decomposition provides an important contribution, especially in terms of phosphorus, but also not negligible in terms of nitrogen pools all over the world. There's a paper, I think, published on PNAS. So, as you can see here, we demonstrated that, in other words, the effects of the carbon released by viral decomposition. So the viruses die, and they live maybe a few minutes when they're free out of the cells and in the marine environment. And if they don't find another host, they actually degrade, and they provide an important contribution in terms of even 0.1 gigatons of carbon per year, that are equivalent to 7% of the carbon, or the organic carbon fluxes associated to the input from the water column, 12% of the fluxes of organic nitrogen through the water column, and even 24% of the phosphorus fluxes in the water column. So, in other words, the viral detritus can contribute for up to 24% to the diet in terms of organic phosphorus for the deep sea life. So this is given a better picture linking, biological components, interactions between different life forms with the functioning of the ecosystems, trying to cope with the gaps of knowledge that indicate basically should be impossible to live and to work at that rates in the deep sea. Now we are understanding a little bit more about the engines of this, functioning, and we are looking a little bit more to the role of this extracellular DNA, understanding the double, implication of that. From one side, we demonstrated trophic reserves of extracellular DNA, but from the other side, keep in mind that the DNA does not disappear immediately before being degraded. Part of the DNA is broken down into small pieces, into small genes that can be used by other microbial organisms, giving them some new functions, new competencies, and even potentially mobile elements that can go into a dynamic evolution of the microbial components. So, coming back, this is a summary of what we said already in terms of carbon to phosphorus ratio that is the highest in the DNA than all of the components. And these are the calculations we made about all biogeochemical implications, which are very important, especially in the top maybe 8 to 10 centimeters of the sediment surface, where most of the life forms are present in terms of also larger components. And these are the biogeochemical implications of extracellular DNA. You can see here the role of extracellular DNA contribution to the prokaryotic metabolism. In other words, we understand very well that DNA is used by microbes. Extracellular DNA is used by microbes, and that component alone can contribute for 47% of the food requirements of deep sea microbes. So extracellular DNA that is largely produced by and released by viral infection and the composition of detritus represent an important source sustaining prokaryotic metabolism in the deep sea, and not only prokaryotic metabolism in the sea. And you can see here the interactions between viruses, prokaryotes, and other life forms, and the release of viruses that infect in the cell lysis, create the loop in terms of either trophic source, but also potentially gene release and inclusions of this loop to change the diversity and the gene portfolio of some microbial components. We also look at the genes containing extracellular DNA. We're looking to different 16S DNA sequences and looking at the components that were contributing to these extracellular and intracellular DNA pools. And I'm not going to much deeper on that, but now we have some molecular tools that can really provide unprecedented information about everything. Now, the last evolution of these studies that we started with the first paper in Science are the so-called environmental DNA. You maybe noticed that this new definition. The environmental DNA is, to my opinion, a wrong definition. The correct one should be extracellular DNA. But the environmental DNA is overall all DNA you can find in a system. 90% is extracellular, 10% is associated to life forms. And you get a bulk of DNA, you do molecular stuff on them, and you understand everything that is there, or was there and is dead, or wasn't there but was imported by another system, possibly as a detritus. Another issue was another intriguing one. How many life forms are there? How many species are there? We don't have clear estimates, even for the large components. Craig knows very well that the effort of the census of marine life from 2000 to 2010 tried to provide some estimates. We are still in the middle of nowhere, in the sense that possibly some 250,000 species are being quantified in terms of multicellular organisms. But the estimates of the diversity in terms of larger life forms range from 3 millions to maybe 8 millions or something like that. The point is that we have no idea at all of the microbial diversity, and neither in terms of bacterial diversity, archaeal diversity, and viral diversity. So we try to use the most recent approaches also to estimate the global viral diversity. It's enormous work, but just to give an example, we found completely different viruses. Keep in mind that 1 kilogram of shallow water sediments contain 10 to 7 viral types. Okay. Of course, the global viral diversity is much higher, but we have no estimate so far. Is highly diverse the viral diversity everywhere. Look at also at the evenness or the classical index of diversity, like Shannon-Weaver. This is the paper by Breitbart, I think was published on PNAS, and these are some other estimates we made from different systems all over the world. So high benthic viral diversity everywhere. Of course, there are some differences in the North Atlantic, more, for instance, than in the North Pacific. At least in the few samples we were able to collect of deep sea sediments, of course. And also within the same groups, for instance, looking at the group of Tamarchiota, there are completely different clades in different deep sea ecosystems. So even within the same group, the same phylum in terms of, for instance, archaea, completely different, there's a clear biogeography also for microbial components. So this is clear for benthic viruses, for benthic archaea, and it's clear also for benthic bacteria. And there are different nucleic acid types, double strand, single strand, DNA, single strand RNA, and so forth. So, conclusion, huge amount, uncounted yet number of viral taxa, say, in the deep sea. And also completely changing, completely different viral assemblage composition in different worlds. We are now exploring the viral diversity in some trenches with a project of Ronnie Glad and others, and Matthias Middelbo. There is also biogeography in terms of viruses in the deep ocean. And you can see here, different ways to visualize different families and groups of viruses that change in all systems that we investigated. And here you have another representation of that. This is, of course, associated to different putative functions that can be highlighted in terms of genes working on amino acids, carbohydrates, fatty acids, metabolism of aromatic compounds. Viruses are extremely plastic and able to incorporate and retain also part of the genome of their hosts or the bacteria, the archaea they infect. But it was clear, in any case, that this huge diversity has a role, plays a role, and the role is keeping control the abundance and the growth of bacteria and archaea, and also keeping high their diversity through a mechanism that is equivalent to the intermediate disturbance hypothesis. Now let me go to the last part of the talk about what is going to happen in the future. What is going to happen in a deep sea, in a changing deep sea that is changing like any other system. We assume, of course, that changes are slower in the deep ocean, and they are indeed, but we have also to take into account that even a minor change in the deep sea is long-lasting, acts at very huge spatial scales, and impacts on life forms that are not used to any temperature or environmental shift. They are used to generally to very constant conditions. And the changes we are showing in the deep sea are huge. Just to make an example, Betou was a French colleague that was exploring and publishing in Science a raise of 0.16 degrees in 30 years. It was doing the PhD work in the 60s and then coming back in the 90s, and the deep sea temperature in the northern Mediterranean Sea increased by 0.16 degrees. Can be huge, can be modest. The fact is that in the next 15 years, so half of the time, the same shift occurred. So another 0.15 degrees of temperature increase was observed. So the deep sea is warming, is progressively warming with important impacts that we also try to evaluate and assess together with Moriaki Yasuhara quite recently. And with Sweetman and others, we also modeled changes in the deep sea floor, look at the projections of the IPCC by 2100, look at the terms of temperature, dissolved oceans, so increasing temperature, decreasing dissolved oxygen, of course decreasing pH, and decreasing the POC, particular organic carbon fluxes, to the deep sea floor. And the point is that the deep sea is not just facing one problem. It's facing all of these problems, plus the direct human impacts, infrastructures, trawling, bottom contact fisheries, or whatever, at once. And these effects act synergistically. which is the expected response of different components. Well, looking at the macro scale distribution of some components, for instance, Tamarchaeota, Eurarchaeota, among here, looking at different, links between the response of bacterial abundance versus the amounts of bioavailable carbon pools, looking at the trends of at the patterns, sorry, of changing bacterial and archaeal abundance at different bottom temperatures and so forth. It is evident one single feature that we published recently on Science Advances, that temperature can change significantly the relative importance of bacteria and archaea appear to be far more sensitive than bacteria to changing temperature, and particularly to rising temperatures. So let's say in the equatorial sediments, there is a higher percentage of bacteria than archaea, far higher, but and this is common in all sediments of the world, but this is even more evidence than at the poles because of the higher temperatures. But if you look at the future in which deep sea sediments warm up, then the archaea are expected to increase in their relevance, and of course to change the functioning also of deep sea ecosystems, because of course this will have an impact on the metabolism of bacterial archaea. And in the global picture we can say that we will see a decreasing input of organic carbon to the seafloor, while the heterotrophic and chematotrophic components will increase their demand. In other words, will increase the competition for organic carbon between microbial components and benthic invertebrates or larger animals. So there will be an increasing problem for the larger species, for the macroscopic species and macroscopic life forms, because the food shortage will be even stronger. So given a picture, a complex picture of what is happening, we have to understand that until recently, we were just focusing on the big things, on the microbial diversity. So interaction, megafauna, macrofauna, then more recently meiofauna, but all these life forms were, assuming to be, interacting with the ecosystem, functioning, delivering ecosystem services, producing biomass, species of commercial interest or whatever. The missing points were the actual interaction between microbial components and these components, protists that are still little known, and the viral diversity. And it looks like viruses interact with all of these components because they infect microbes, they infect protists, they infect all of metazoans, and they contribute to the global biogeochemical cycle. So if we want to have a better picture, we want to make a picture that considers all of these components, we have to understand in the future we should not just look at interactions between big things, but also between big and small, and in different ways. Maybe microbial components can compete, maybe can be a food source, viruses maybe can impact on big forms, but also can reduce the abundance of microbes, thus leaving more food for large forms. So multiple interactions that can switch according also to ongoing climate change. So one curious question could be, but can viruses save the planet? Well, we don't know yet, but it's certain, but it's clear that they can certainly affect or influence the overall functioning of the oceans in many ways. We understood that they can certainly change, for instance, the export of organic matter to the deep sea floor, because the higher the infection of viruses in the shallow waters, and the lower the amount of POC that will reach the deep sea floor. Why? Because if you lease the cells, the release of dissolved organic matter will hamper big particles to get down. No? The larger the particles, the faster and the more relevant in quantitative terms the input of organic carbon to the deep sea floor. But if you through a viral shunt you kill everything up there, microbes, copepods, rather than phytoplankton, then very little can reach them down there. Then look at the deep sea and look at the increase of temperatures. What happens with the interaction between microbes and viruses? Can viruses release DOC and stop the growth of bacteria and other prokaryotes, thus leaving some more food for the remaining life forms in there or not? Viruses can certainly be big players in that equilibrium. And finally, it's not related to the deep, but think the importance on the climate regulation. We assumed for many years that, for instance, clouds can be also dependent on the cloud condensation nuclei that are produced by, we thought many years ago they were just salt crystals, some NaCl salts, marine salts. And then we discovered more recently, in several papers published on Nature, that there are small organic particles creating cloud condensation nuclei, so creating the conditions for cloud formation. Now we understand that also viruses released in the aerosol can contribute and can represent cloud condensation nuclei and can possibly regulate that. So viruses can infect the algae, can be released into the atmosphere, can increase the cloud formation, can reduce irradiation, can reduce dust production, phytoplankton production. So they can also play a role in regulating the climate through the regulation of the cloud formation. So a couple of conclusions about that. The first is that we have a virusphere. We have a big microbial world. In current research, we are giving important attention to the microbiomes, so the association of microbial components to the large life forms. But there is also a virusphere, a world of viruses that interact with all other components with a huge diversity and important contribution to life and death in the oceans and the deep sea, and ubiquitous components sensitive to many variables, including temperatures, including acidification, including changes in organic carbon fluxes, so that this is another big picture that is available on recent papers on CAM biology. We should try to look at all these components together, looking at the role of viruses in affecting all components, these tiny dots, gray dots there. There are a lot of bacteria in playing important roles for releasing, producing organic matter. In the circulation, the effects of global change in food circulation, oxygen, temperature, pH, that is affected differentially all of these components singularly and collectively through the alteration of the interactions between these components. To conclude, I hope I'm not too long, I hope I've given an overview, quick, maybe too quick and too rough overview of this invisible living planet. To my opinion, viruses are some magic players. They are typically seen as our enemies, killers of life, but they're far more than that. They are needed to sustain life forms, to sustain the cycles, the biogeochemical cycles of the planet. They can also define winners and losers. They can shape ecosystem. They can push for evolution. They can control food webs and regulate even the climate. and sometimes they might be even our best friends in controlling some processes that need a regulation. Of course, this is not the case of SARS-CoV-2, or not, because maybe this tragedy of the pandemic COVID is killing a lot of people, but maybe is also giving us the clear perception that there are strong interactions between our planet and ourselves. And we need to respect and protect the planet, because abetting biodiversity, destroying natural systems is at the beginning of all the story of the zoonosis, the transmission of pandemics from wild animals to humans. Has been that in the past, is presently the same with the COVID-19, and will be also in the future if we do not do more for preserving the marine ecosystems. So thank you all for being with me for this hour. Thanks to my colleagues, Cinzia, Antonio, Pusceddu, Delano, Rastelli, my lab, and of course thank Craig. Thank you very much, because I was following your seminars even before your invitation, and it has been an honor for me to be with you and to contribute to your class. Thank you very much.
Thank you very much, Roberto. Let's go into gallery view so that Roberto can see all of us applauding for a very nice talk. I think it gives us a much broader perspective about the world than we've had in any of our other seminars. So thank you very, very much. Are there questions? Sebastian.
So one of my questions is, how do you differentiate between viral and bacterial DNAs in your gene samples, and also determining between organism genomes? How do you know where one gene starts and the other gene ends, or whether they're fragments or stuff?
Yeah, this is a good question. This is basically related to the sequences that are, specific for different, components. This is more and more evident as we increase the databases having sequences assigned to any form. Of course, I have to be honest, there is a certain degree of uncertainty in some cases, because we are progressing enormously in terms of bioinformatic, analysis, computing. So putting together these, sequences, recombining and creating, the full structure. But often it might be the case we create chimerae, strange, Frankenstein genes, pulling together. So it is an approach, trials and errors. We are progressing, but we are not sure. And this is not for only for microbes. We try the same approach for barcoding of benthic fauna, for meiofauna. We publish papers on that too. And the problem is that you have the sequence, but then you don't know to which species they belong to, because there are no sequences in the databases. So you got a point that is crucial for the future. And that indicates that one of the most important positions in the future will be also the one of the expertise in bioinformatics. At present, the biologists, specializing in bioinformatics, which is like biology, is everything. Bioinformatics is not just one thing. It's a huge field, because there are those specializing in microbes, in viruses, in plants and whatever, but the bioinformatics is growing as a sector, and we are searching for bioinformatics, because everybody needs to understand more about this big data produced by massive high throughput sequencing. This is a big problem.
Thank you. Anybody else have a question for Roberto?
Sure. George, maybe?
I asked a previous lecturer, why is it that we see so many invertebrate forms and let's think about jellyfish and comb jellies and so forth. And we see them only in a moment in time, but it seems that there's not much infectious disease among many simple creatures. Think of all those hydroids and so forth, corals. How can it happen that they resist overwhelming bacterial and viral destruction?
Yeah, that's is a good question. I think in some cases we just don't know. Until a few years ago, there was no evidence even on viruses infecting diatoms, and then we discovered that diatoms are infected. And so there were, theories about why diatoms, because they have TK, silicious, TK, whatever, and then we discovered that they are infected too. How they can resist, we don't know if they resist actually. Certainly, viruses need host to grow and need host to infect. So these organisms that are larger but more intersparse and far each other can be less susceptible to infections because they're simply, they stay apart. They don't even in some cases touch for reproduction, just release the gametes and eggs and whatever and the environment. So they reduce the potential contact rates. So viruses depends on the ability to get in touch with the hosts. So if they are a little bit far, maybe this is a good solution. And this is the same that is happening among us in this period. Stay apart so that we don't pass each other the virus, maybe. Not sure I responded to your question, but it's,
Thank you.
You're welcome.
George.
Hi, I'm curious if you have an estimate of any kind about what fraction of the quantifiable virus sphere represents fluxes that take place in, for example, free-living prokaryotic populations versus commensal and or endosymbiotic concentrations of prokaryotic. I guess I'm thinking specifically about the phage and prokaryotic hosts, and what fraction is it a minuscule fraction that takes place in, for example, symbiotic populations, or is it a big chunk? Do you know?
Yeah, no, I don't know. Not exactly. I know that this is a very tricky question because indeed, sometimes viruses are our friends, and we also have, our genome retroviruses of previous infections that remain through, because the non-lytic, so the lysogenic or the latent infection, so the viruses that injects its genome in the host, but then doesn't complete, the reproduction and the lytic cycle, protects the host from other infections. It's like a vaccine. So it looks like most of the populations of microbes, and likely also of other, invertebrates, have a latent or non-lytic or lysogenic infections that are a system to protect them from other infections. But for instance, we demonstrated on corals, we published some papers on, for instance, the impact on some substances. You have a lysogenic infection, so you are protected by other, maybe, nasty viruses, and then something like UV, like hydrocarbons, heavy metals, or even a sunscreen, switch on the lysogenic infection into a lytic virus, into a lytic infection, and releases the viruses and kills the host. So it's a delicate equilibrium in between, what is the kind of, not really of course symbiotic, because this is a pathogen, but the coexistence for mutual benefits. The beneficial effect for the infected, organisms, is they're protected somehow from other infections. The beneficial effect for the host is that they might kill you, but they are not sure they can find another host, so they can die when they really are released. So better to stay latent, lysogenic, underexpressed in the host than being winner, killing, and die because they don't find another host. So this is another solution that evolved through life. But it's a very complicated question, so I'm not sure I've been able to respond correctly.
Others? I have a question, Roberto. There's this vast literature about control of communities and ecosystems, and people work on predation and competition and all of these factors that kill organisms or change their distributions and so on. How important is it to include viruses? Do viruses directly kill very many of these larger organisms? And if so, should that be incorporated into our thinking of how communities are regulated and populations are controlled and so on?
Yes, Craig. I think we know too little yet. But for sure, for instance, if you look at the case of at the hecatombs of marine mammals, for instance, since maybe 2 decades is known that Coronaviridae, so the same family of the SARS-CoV-2, is infecting sea lions, dolphins, sea whales, determining massive strandings of even more than 100 individuals, even in the US coasts, that were attributed definitely to these viral infections. So just to give an example of the Coronaviridae, because now we are facing the COVID pandemics, this is present, the Coronaviridae family is present in marine ecosystems and can kill a lot of life forms. Of course, this is a case for mammals. By the way, exactly with the same symptoms, lungs, whatever, viruses do the same for all life forms. So the problem is that we don't know that exactly. We started looking at the infections of microbes because this is closer to the field of the marine microbiologist, environmental microbiologist, while the infections of animals are related to the veterinarians. So it's a little bit, is a cross discipline. So now we understand that, for instance, parasites are crucially important for, ecosystem equilibria. In the future, we'll understand that also viruses and other pathogenic forms are important for other forms. We understand a little bit better, for instance, for coral disease and related to many prokaryotic infections, bacterial infections. Soon we will understand more about that, but I'm confident that this certainly possible that viruses play an important role, as for humans, also in the control of all life form populations, including cephalopods or worms, polychaetes, nematodes. Nematodes for sure, because nematodes are 350 of the metazoans on Earth, so they are very abundant. So, yeah, I think so.
Okay, I'm looking at the chat room here. I think Aaron Galloway has a question. Aaron, would you like to ask yours?
Sure. Thank you. That was an amazing talk. I learned so much. As a kelp, kind of nearshore marine ecologist, I have my kelp forest here behind me, Roberto. I'm often trying to put in context the various kind of components of primary production and what they mean to each other. And of course, chemosynthetic production matters. And one of the things I've always struggled with is trying to, well, what I'd like to ask you, because I have a feeling that you might have a handle on this, is what proportion of the total productivity in our marine biosphere do you, or, kind of when you think about the models that you've talked about and the results and as it relates to viruses especially, what proportion of the total productivity is completely independent of photosynthesis? Do you have a handle on that? And because like when I look into the literature on that, it's kind of overwhelming. There's so much there and I don't know where to start. And so I thought you might have a perspective on that, given your talk.
You mean chemosynthetic primary production?
Yeah.
Or
yeah, kind of just in general, the kind of the dichotomy between light independent and chemosynthetic production.
Yeah, as a guess, because it's not based on a global scale, but I think that 10 to 20% of the global carbon production in the deep is due to chemosynthetic primary production, excluding hotspots like hydrothermal vents or other systems, excluding the chemosynthetic production associated to symbiosis with other invertebrates or life forms. Mm-hmm. So, of course, it's not dominant, but it's not negligible. And I think the more we will understand and the more we'll reevaluate also this component, the chemosynthetic. Yeah.
Thank you.
Now, also, Sebastian, who asked us a question earlier, has written some things in the chat room. Would you like to ask Roberto directly what your questions are, Sebastian?
Yeah. So one thing I've heard recently, or at least maybe a few years ago, that was increasing interest in the viral field, are the giant viruses that have been discovered that are like in between viruses and cells. Do you know of any developments on that?
Yeah, thanks, Sebastian. Very, very timely question. Giant viruses are of growing interest. Basically, is like is a predator-prey interaction. So the smaller the prey, the smaller the predator. The larger the prey, the larger the predator. And giant viruses are, for instance, more relevant than in other systems in Antarctica or in aquatic systems where we have large eukaryotes, large phytoplankton cells. And these are important components also in the ice pack. We are doing a program on giant viruses there. And, but also apparently in other systems and that they can be relevant. They are of course, giant in the sense that they are of the size of a bacterial cell or whatever, but definitely more than classical viruses that are in the order of a few nanometers. So yeah, interesting component, still almost completely unknown.
Okay, thank you. This is your last chance. Does anyone have a final question for Roberto? George, I might have expected that from you. Go ahead.
I wanted to ask you to comment on the extent to which you can presently assess from environmental sequence data the physiological context in which, for example, viral DNA release took place or something like that. How much can you learn from relative transcript numbers and things like that at this point?
Well, not sure can I answer correctly to your question, George. You mean the confidence that you have in the ability to reconstruct the sequence, or?
I guess, I mean, as opposed to simply learning about the numbers of different viral types, different viral species, is it possible to make inferences about, for example, the recent physiological history of a population by looking at things like the relative numbers of transcripts for things like coat proteins versus replication genes, or quorum sensing type transcripts versus, you know, lysogeny factors or something like that?
Well, yeah, possibly, yes. We were not going that deep personally in that kind of, sorry, and we were not going that deep for a simple reason that I think it's possible, to be honest. I'm pretty sure it's possible. At least we can make an attempt to that, for that. But we were so much interested by the ecological interactions that we were giving less attention to the cellular interactions or the coevolutionary interactions. But this is an open field that requires, of course, also the point that we were describing before, So they are mixing up so much their genome with the one of the hosts that sometimes it's very difficult to distinguish between the two. And it is sure, but we have seen that also for forums that were, you know, including in their DNA photosynthetic genes, so in the deep sea. So I mean, the smaller you are, the higher the possibility to combine your DNA and to select the forms that are more adapted to play specific roles. And even the identification of the putative function that is now very, you know, is a topic that is you know, of high attention for scientists, to my opinion, has still a high level of uncertainty. So from my point of view, since I'm not a microbiologist, I look forward to exploit well solid, you know, tools to apply them to ecological questions, than to develop the tools, the methodological tools to go deeper in the problem. And I think we are still a little bit immature to fully exploit that potential and to define all functions of all genes and how these are, you know, not just the mechanism, but the specific use of these genes by different life forms and their, you know, passage from one species to another one. So the viruses contribute in that terms, either, you know, taking up and either reinfecting, passing genes from one species to another. Because coming back to another ecological concept is that viruses, through the mechanism that is killing the winner, they infect first the most adapted, you know, species, because they are dominant, they are very abundant. So for the contact rate, they infect more easily those that are abundant, right? Then they get into the DNA, maybe they pick up some genes that statistically are those that make this species more adapted to that conditions. So they kill the strong, like Robin Hood effect, okay? They kill the strong, and then they infect another one that maybe is not so abundant because it's not well adapted. But then they pass into the new host the genes that provide adaptation to the best conditions. So they take to the rich and give to the poor. So they are also able to, act as a kind of, I call that Robin Hood or Zorro effect. I don't know, it depends on your, on your hero, right? But the effect is that viruses can contribute to make more equal the ability of all hosts to face the environmental conditions and to give them the opportunity to adapt. So this is not a response to your question, but also to give you an idea of the complexity of the mechanism of infections in which you are not just killing, but you're killing one, but then taking something, moving to another one, like a mosquito, you know, that is infecting, spreading malaria, just, you know, or parasites or disease. So from one to another. So.
Okay, well, thank you very much, Roberto. Incredibile, grazie. And let's give him another round of applause. And Roberto, you can go eat a pizza margherita or something now.